6. Genetics and molecular biology. Chromosomes and how they are composed affect where and how DNA is accessible in genes. One type of mutation are nucleotide pair substitution mutations. These mutations can be silent, which have little to no effect and are the same amino acid. Missense mutations, which are a different amino acid but little to no effect, or nonsense mutations, which are a different amino acid and have an effect. Another type of mutation are nucleotide pair insertions or deletions. These can be frame shift mutations, which can be detrimental to the folding of the protein and what is produced. This is because a mutation that is not in a set of 3 can affect the rest of the sequence and therefore affect what is transcribed. Like in bacteria, asexual reproduction occurs. Asexual reproduction is a type of reproduction by which an offspring arises from a single organism, and it inherits the genes of that parent only. It does not include the fusion of gamete cells. Sexual reproduction does include the fusion of gamete cells, which are each haploid and then together form diploid cells. This is a much different process, and crossing over can occur during chromosomes to increase genetic variation in sexual reproduction. Mitosis and meiosis are very different as shown in the picture. Mitosis can be used in asexual reproduction, and meiosis is used in sexual reproduction. When chromosomes do not split in a typical way, aneuploidy can occur, which can lead to genetic disorders like trisomy 21, which is when you have 3 copies of your 21st chromosome. Additionally, monosomic disorders could occur as a result of non-disjunction. There are 2 different laws that describe patterns of inheritance. The first is the law of independent assortment, which states that genes are not linked when inherited. The second law is the law of segregation, which states that 2 alleles for a gene separate during gamete formation. Punnett squares can be used to predict the genotypes and phenotypes of particular crossbreedings of species or different alleles. As shown in the diagram, brown is the dominant allele and blue is the recessive allele. So either being homozygous for a big B or heterozygous. Heterozygous allows you to have brown eyes, while you must be homozygous recessive to have blue eyes. Punnett squares can be used when determining simple dominance, incomplete dominant, codominance, multiple alleles, sex-linked traits, and polygenic inheritance. Genetic engineering and its research includes many things like restriction enzymes, plasmids, and gel electrophoresis, which is used to determine DNA fragment lengths in genetic engineering. While some genetic engineering principles are already being applied in different places around the world, genetic engineering poses many ethical questions.